Hello student, all of you are most welcome on my channel, that is Liberty Solution. In today's video, I will be discussing about class 12th biology, chapter number 2, reproduction in flowering plant. Like, comment, share it with your friend, and don't forget to subscribe my channel to get many more videos. These videos are useful for your board examination, school examination, as well as NEET examination. So let us start our chapter. So we'll be discussing about that chapter number 2, sexual reproduction in flowering plant. That is sexual reproduction in flowering plant. Flowering plant means angiosperm. We have discussed about in class 11 what are thallophytes, bryophyte, pteridophyte, gymnosperm. And till gymnosperm, there is no flower. That's why they are known as non-flowering plant. Flowers are was visible only in angiosperm. That's why they are called also known as angiosperms are also known as flowering plants. So all flowering plant, that is angiosperm, they show sexual reproduction. There is what sexual reproduction in flowering plant. And flowers are the sites of sexual reproduction. So all the angiosperm plant, they show sexual reproduction. And what are the site of sexual reproduction? This flower. Flowers are site of sexual reproduction. Like this is a China rose. This is a flower. Flower is the site of sexual reproduction. See, several hormonal and structural changes result in differentiation and development of floral primordium. This is what floral primordium. So here there are different types of primordium. Carpel primordium, sepals, petal primordium, stamen primordium. So these primordium bears different organ of the flower. Now there is inflorescence. Inflorescence bear the floral buds and then the flower. We have already studied what is inflorescence in class 11. Arrangement of flowers on the floral axis, that is called inflorescence. Racemose, cymose, we have studied in class 11. So in the beginning, flower is in the form of what? Floral bud. And then this bud, this is a what? Floral bud. And ultimately this bud opens and then it is converted into flower. Now structure, will be discussing about structure of flower. These all are the events are what related with pre-fertilization, structure and event. So a typical flower has two parts, two sexual parts. One is what? Enroutium and second is what? Gynocim. These are what? Reproductive parts. There is a calyx, corolla, but they are not reproductive part. So Enroutium and Gynocim, they are what? Main reproductive parts of a flower. So this is what? This is a flower. Here there is Enroutium. This is a bisexual flower. Why? Because both male sex organ Enroutium and female sex organ, that is Gynocim, is present together. This is a Gynocim. Gynocim is also known as carpel. Enroutium is also known as what? Stamen. If we see, Gynocim is divided into stigma, style, and ovary. Inside ovary, there are ovules. They are nothing, they are what? Megasporangia. If we see Enroutium or stamen, it is divided into two parts: filament, this is filament, and anther. Anther is having microsporangia. Gynocium, yeah, Gynocium is having megasporangia, and anthers, they are having what? Microsporangia. Megasporangia are present in ovule region, and microsporangia are present in anther region of stamen. Within microsporangia, there are pollen grains are there. So if a typical flower is having two parts, one is what? Enroutium, second is what? Gynocium. I hope it is clear to all. Let us proceed. Now we'll be discussing about Enroutium. It is a male reproductive part of a flower. So Enroutium is what? It is also known as stamen. It is what? Male reproductive part of the flower. It consists of whorls of stamen. Their number and length are variable in different species. So here, this is a stamen. It is having what? Microsporangium. It is made of two parts, filament and anther, and within anther there is microsporangium, and within a microsporangium, microspores are produced. If you see this flower, here there is what Gynocium is there, that is stigma, style, and ovary, divided into three parts. Then there is what perianth is there. Perianth means petal and sepal combined together. They are known as what? Perianth. Then this is a floral axis. This is articulation. This is a pedicel. These are nectary. There is a deposit of nectar. So now we have learned that there are two parts of stamen. One is what? Filament. Second is anther. This is filament. This from here, this is a filament, and this one is what? Anther. Now we'll be discussing about the filament. Filament is long and slender stalk. This is long, slender stalk. Other name of filament is what? Stalk. It is proximal end is its proximal end is attached to the thalamus. So this is proximal end. This is distal end. Proximal end is connected with what? Thalamus or the petal of flower. So these are they are attached with petal or the thalamus. If it attached with here, that is called thalamus. If it is attached with petal, so epipetalous. Terminal and typically bilobed. Anther now, what is anther? It is terminal. It is present at the top, that's why terminal. It is bilobed. See here, it is bilobed structure. One lobe, two lobe. It is what? Bilobed structure. Each lobe has two theca, diathecous. This is one lobe, and this is second lobe. So each lobe is having two theca. This is thickening. That's what it is known as diathecous. Often a longitudinal groove run lengthwise in separating the theca. There is what? Longitudinal groove. It separate these two thecas. Now we'll be discussing about transverse section of anther. This is a stamen is divided into filament and anther. If we are cutting this anther in this way, horizontal way, we'll be getting transverse section. We are cutting in this way, we'll be getting what? Longitudinal section. So this is what? Transverse section, and this is what? Longitudinal section. So we'll be discussing about transverse section of anther. So the anther is a tetragonal structure. There are what? 4 cones are there. 1, 2, 3, 4. Corners are there. Therefore, tetragonal structure consisting of 4 microsporangium located at the corner, 2 in each lobe. So there are what? At the corner, there are what? Microsporangia are located. So this is microsporangia. This is 4 microsporangia are there. 2 microsporangia in each lobe. This is one lobe. This is second lobe. So here in this lobe also there are 2 microsporangia, and in this lobe also there are 2 microsporangia. The microsporangia develops 2 pollen sac. This microsporangia develops into pollen sac. These are what? Pollen sac. Microsporangia develops 2 pollen sac. They extend longitudinally all through the length of an anther and are packed with pollen grains. So within pollen sac, there are what? Pollen grains are there. These are pollen grains. These are what? Pollen. Now, structure of microsporangia. A typical microsporangium is near circular in outline. If we see this is a microsporangium, it is what? Circular in outline. This is circular in outline. This is what? Circular in outline. Each microsporangia is surrounded by 4 wall layers. What are they? Epidermis, endothelium, middle layers, and tapetum. Tapetum is the innermost and epidermis is outermost. So epidermis, endothelium, middle layer, and then tapetum. 4 layers are there. Here, this is epidermis followed by endothelium, then middle layer may be 2 or 3 layers. Then there is what? Tapetum, innermost layer, that is called tapetum. So this question will be there in the CBSE board examination as well as competitive examination. Arrange this layer from outside to inside, yeah, inside to outside, yeah. What is the sequence of this layer? So epidermis, endothelium, middle layer, tapetum. The outer 3 layers give protection and help in dehiscence of anther to release the pollen. So these first 3 layers, they help in what? Their function is what? To protect the pollen grains. And they also help in dehiscence of anther to release the pollen grain. The tapetum, that is innermost layer, nourishes the developing pollen grain. So this layer, tapetum layer, it provides nourishment to developing pollen grains. Cells of tapetum contain dense cytoplasm and generally have more than 1 nucleus. They may be having more than 1 nucleus. In young anther, each microsporangium has sporogenous tissue. They are having what? Sporogenous tissue. This is what? Sporogenous tissue. Inner to the tapetum layer, there is what? Sporogenous tissues are there. It consists of compactly arranged homogeneous diploid cell. They are what? Diploid cell. These are what? Sporogenous tissues. They are made up of compactly arranged homogeneous. Homogeneous means similar origin. All cells are diploid cells. They are called sporogenous cells. Now microsporogenesis. What is the microsporogenesis? So as the anther develops, each sporogenous cell, that is called microspore, or pollen mother cell. So here there are so many cells. Inner to tapetum, there are sporogenous tissues. These sporogenous tissues are made up of cells. So one cell will be working as what? Pollen mother cell. That will be diploid in nature. So that is known as PMC, a pollen mother cell. This pollen mother cell undergo meiotic division, meiosis division. That is what? A reductional division to form microspore tetrad. So this is what? Microspore mother cell, which is diploid in nature. Diploid means 2n. It is diploid in, it is having two set of chromosome. So it will undergo which division? Meiosis, not mitosis. It is will undergo meiosis, and then ultimately from one cell, 4 cells will be produced. Okay. So that is called what? Tetrad of pollen. These tetrad in the beginning they will be together, so but after some time they will get separated. So from one pollen mother cell, 4 pollen grains are being produced. These are four pollen grains. The formation of microspore from a pollen mother cell PMC through meiosis is called microsporogenesis. So what is microsporogenesis? The process of formation of pollen grains from pollen mother cell by meiosis, that is called microsporogenesis. Now we are doing what? Microsporogenesis. As the anther mature and dehydrate, this anther will become mature and this will get what? Dehydrate. The microspores dissociate from each other and develop into pollen grain. Then ultimately microspores develop into pollen grain. Each microsporangium contains thousands of pollen grains. They are released with the dehiscence of anther. Dehiscence is nothing, it is what? Splitting up anther. That is called dehiscence. So this is one mature dehisced anther. Now we'll be discussing about pollen grain. What is pollen grain? Pollen grain is also known as male gametophyte. It is generally spherical in shape. This is pollen grain that is spherical in shape. Diameter is 25 to 50 millimeter in diameter. Cytoplasm is surrounded by a plasma membrane. A pollen grain has two-layered wall, XII and intine. It is having two layers. That is, one outermost is XII, and then inner one is what? Intine. It is having cytoplasm. It is having nucleus. It is having germ pores. There are 2 cells. One is what? Bigger cell, that is vegetative cell. Smaller cell, that is generative cell. So this is a structure of a mature pollen grain. So sometimes the question will be there, draw a well-labeled diagram of mature pollen grain. So what you have to show 2 cells. One is what? Generative cell is smaller, and then there is vegetative cell. These cells are having single nucleus. Then there is inner layer, that is intine, then external layer, that is what? XII, and then at certain position there is what? Germ pores are there. Now, the hard outer layer made up of sporopollen. This XII is made up of one chemical that is called what? Sporopollen. Very frequently asked question, very popular question. So in XII of the pollen grain is made up of sporopollen. It is highly resistant organic material. It can withstand high temperature. It can withstand high temperature and strong acid and alkali. Enzyme can degrade sporopollen. XII has aperture called germ pores, where sporopollen is absent. In germ pore region, there is no, like these are germ pore. In this region, this XII will not be having sporopollen. Pollen grains are preserved as fossil due to the presence of sporopollen. XII exhibit patterns and design also. See, there are different pattern and designs, and this design is because of sporopollen. Now we'll be discussing about intine. It is inner wall. It is a thin and continuous layer. It is made of cellulose and pectin. It is not made up of, intine is not made up of sporopollen. Xine is made up of sporopollen. So intine, what is chemical nature of intine? It is made up of cellulose and pectin. It is a carbohydrate. Cellulose is a carbohydrate. Pectin is a protein. A matured pollen grain contains two cells, vegetative cell and generative cell. This is what, how many cells are there in mature pollen grain? It is generally having two cells, generative cell and vegetative cell. So vegetative cell is a bigger, has abundant food reserve and a large irregularly shaped nucleus. Generative cell, it is small and floats in the cytoplasm of vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. Over 60% angiosperms shed their pollen grains at two-celled stage. In other generative cell, divides mitotically to give two male gametes, thus pollen grains are set at three-celled stage. This generative cell will divide and it will produce what, two male gametes. So ultimately, pollen grain will have what, three-celled stage. Now, Enroutium, we are doing what, Enroutium, pollen grain, male gametophyte. The seed pollen grain have to land on the stigma. These are pollen grains are released from the here, Enroutium, stamen, from Anther. Now, these have to land on the stigma before they lose viability. The viability period of pollen grain is variable. It depends on temperature and humidity. That means life. So lifespan of the pollen grain varies in different plants. Okay, and it depends on temperature as well as humidity. So viability of pollen grain of some cereal like rice, wheat is 30 minutes. Some members of Leguminaceae, Rosaceae, Solanaceae have viability for a month. Now we'll be discussing about economic importance of pollen grains. These means pollen grains are rich in nutrients. Pollen tablets are used as food supplements. Pollen tablets and syrups increases performance of athletes and race horses. They are stored for a year in liquid nitrogen at the temperature of minus 196 degrees Celsius. They are used as pollen bank in crop breeding program. Pollen grain of some plant like parthenium or carrot grass are allergic for some people. It leads to chronic respiratory disorder called asthma, bronchitis, and many more diseases. So this is a parthenium plant. That is called carrot grass. It is a seaweed. It causes allergy. It is a weed. It is not seaweed. It is a weed. It causes allergy. Now we'll be discussing about Gynocim. Female reproductive part is called what, Gynocim. It may have a single pistil. A plant may have single. In Gynocim, there may be single pistil, or if there is in Gynocim, one pistil is there, then that is called monocarpellary, or more than one pistil, then multicarpellary. In multicarpally, the pistils may be fused together. If pistils are fused together, that condition is called syncarpus. So here, the pistil, there are so many pistils, 1, 2, 5. 5 stigma means 5 pistils. These pistils are fused together. So syncarpus. If pistils are free, that is called apocarpus. These carpels are free, not fused. Apocarpus. So hibiscus shows what? Syncarpus. This is what? The multicarpally syncarpus pistilla power, somnifera. That is, here it is what? Syncarpus. So sometimes question will be the pistilla power is syncarpus or apocarpus. So you have to write what? It is what? Syncarpus. Multicarpally, Hippocarpus, Gynocina, mycelia. This is what? Mycelia. Here it is what? Multicarpally. And the carpels are free from each other. So it is what? Apocarpus. Each pistil has 3 parts. See, this is a pistil. It is having 3 parts. What are they? Stigma, style, and ovary. What is stigma? Landing platform for pollen grain, that is called stigma. These are what? Stigma. This is stigma. This is the landing platform for what? Stamen. That is called stigma. What is style? Elongated slender part beneath the stigma. Beneath the stigma, elongated part, that is called what? Style. Basal bulge part, that is called ovary. So this bulge part is called what? Ovary. So a gynocium is having 3 parts: stigma, style, and ovary. Then inside the ovary is the ovarian cavity. This is a inside ovary there is what? Cavity. This cavity is called ovarian cavity or locule, in which placenta is located. Inside locule there is what? Placenta. These are what? Placenta is located. Arising from placenta are ovules, that is megasporangium. So these are placenta. From this there is some ovules are attached. So that is called what? These ovules are nothing, they are what? Megasporangium. They are nothing but what? Megasporangium. Number of ovules in an ovary may be 1, like wheat, paddy, mango, too many like papaya, watermelon, orchid, and many more. Gynocium, this ovule is attached to the placenta by a stalk. Ovule is attached to the placenta by a stalk. So this is the ovule. This is a placenta. This is ovule. This ovule is connected with placenta by stalk. This space is called locule. Junction between the body of ovule and funicle is called hilum. So this is what? Ovule. So junction of between the body of ovule, this is whole body of the ovule, and funicle is called hilum. This is what? Hilum. Each ovule has 1 or 2 protective envelopes, that is called integument. So this is ovule, it is having outer integument and inner integuments, except at the tip where a small opening micropyle is present. So here in this region, where that is called micropyle, in this region, this outer integument as well as inner integument, they are not continuous. So this opening is called what? Micropyle. Opposite the micropylar end is the chalazal part, that is called basal part. This is micropylar, this is what? Chalazal. That is part basal part. Enclosed within the integument, there is a mass of cell called nucellus. After inner integument, there is a mass of cell that is called nucleus. Its cells contain reserved food material. Inside the nucleus is the embryo sac. Inside the nucleus, what is there? This is what? Embryo sac. That is nothing, that is what? Female gametophyte. Embryo sac is what? Female gametophyte. Like pollen gland is what? Male gametophyte. Here, what is embryo sac is considered as female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Now, Megasporogenesis. It is the formation of megaspore from megaspore mother cell. So what is the megasporogenesis? The process of formation of megaspore from megaspore mother cell, that is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell in the micropolar region of the nucleus. It is a large cell containing dense cytoplasm and prominent nucleus. So megaspore mother cell is a large cell. This is megaspore mother cell. It is very large cell. It is containing dense cytoplasm. If we compare with this neighboring cell, it is having what? Dense cytoplasm. And prominent nucleus. This nucleus is prominent, means bigger nucleus. Megaspore mother cell undergo meiosis to produce 4 megaspores. So this megaspore mother cell will undergo meiosis. So first meiosis, second meiosis. After completion of second meiosis, 4 megaspore tetrads will be produced. So this megaspore, meiosis first produces 2 megaspore, then megaspore, that is called megaspore dad. Then again it will divide, complete meiosis second, then it will be converted into megaspore tetrad. In majority of flowering plant, 1 megaspore is functional while the other three degenerate. So out of these 4, only 1 become functional and 3 will get what? Degenerate. They will get degenerate. So only 1 will be functional. The functional megaspore develops into female gametophyte. Then it will be developing into female gametophyte. So 3 will get degenerate and it will become what? Functional megaspore. The embryo sac formation from a single megaspore is called monosporic development. So out of 4, only 1 is functional. So this type of development of megaspore is called monosporic development. If all are functional, that is called what? Polysporic development. Now, formation of female gametophyte, that is called embryo sac. The nucleus of functional megaspore divide mitotically to form 2 nucleus. So, the megaspore nucleus will divide, it will produce what? 2 nucleus, 1 nucleus 2. This by mitosis. They will again divide by mitosis. So, it will also divide, this will also divide. So, 1 will become 2 and ultimately 2 will become what? 4. Again, they will come divide mitotically. So, there will be 4 at one end and 4 at another end. So 8 cell states. First 2 cell state, then 4 cell state and 8 cell states. These, they will move to opposite poles forming 2 nucleate embryo sac. The nuclei again divide 2 times forming 4 nucleate and 8 nucleated stage of embryo sac. All these things are taking place inside embryo sac. These divisions are free nuclear. Means only nucleus is dividing, cell is not dividing. that is nucleodivision is not followed immediately by cell wall formation. So this is what 2-celled, 4-celled, and 8 nucleated stage of embryo sac, and ultimately this is a structure of mature embryo sac. Mature is for embryo sac is having 3 antipodal cells, 2 polar nuclei, synergids, and then egg. The synergids eggs are present towards micropylar end and antipodals end away from the micropylar end towards salazar. Formation of small gametophyte we are doing. After the 8 nucleate stage, cell wall are laid down leading to the organization of typical female gametophyte. 6 of the 8 nucleus are surrounded by cell wall and organized into cell. The remaining 2 nuclei, polar nuclei, are situated below the egg apparatus in the large central cell. So here there is one in egg is nucleus is there. Again now there will be what formation of cell wall. So there will be what at cell end there will be nucleus. After nucleus dividing nucleus, cell will also divide. It will be producing 3 antipodal cells. Towards micropylar end, there are 2 synergids and 1 egg cell. And then in the center there is what polar nuclei. So the mature embryo sac is having how many nucleus? So a typical mature embryo sac is having 8 nucleate and 7 cell state. How many cell? 1, 2, 3, 4, 5, 6, and this 7. And how many nucleus? 1, 2, 3, 4, 5, 6, 7, 8. How many nucleus are there? 8 nucleus and 7 cells. 3 cells are grouped at micropylar end and form egg apparatus. So what is the location of egg apparatus? It is formed towards what? Micropylar end. These egg apparatus is consist of 2 synergids and 1 egg cell. So what are different components of egg apparatus? Egg apparatus is made up of what? 2 synergids and 1 egg cell. 1 egg cell. Synergids have special cellular thickening at the micropylar tip called filiform apparatus. These synergids are having some of the thickening. These are called what? Filiform apparatus. These filiform apparatus help to guide the pollen tubes into synergids. 3 cells at the chalazal end are called antipodal cell. Chalazal end is having 3 cells. These are called antipodal cells. The large central cell has 2 polar nuclei. This is large central cell. It is having 2 polar nuclei, 1 nucleus and 2nd nucleus. So here there is a meiotic tetrad. There is chalaza. These are integument. These are functional megaspore. Then this is what? Mature female gametophyte. Chalazal side, 3 antipodal cells. Then there is vacuole. There is integument. There is egg cell is there in center. Then egg cells are there. Then synergids are there. These are the arrangement of different type of cell within embryo cell. Now next topic is what? Pollination. Now what is pollination? It is the transfer of pollen grains from anther to the stigma of a pistil. Transfer of pollen grain. This is the pollen grain. Transfer of pollen grain from anther to stigma of a flower. So transfer of pollen grain from the anther to the stigma of the pistil, that is called pollination. Now, what are types of pollination? Based on source of pollen, pollination is of three types: autogamy, ketogamy, and xenogamy. So there are three types of pollination: autogamy, that is called self-pollination, then ketogamy, then xenogamy. So this is what transfer of pollen grain from the anther to stigma of same flower. That is a type of one type of pollination. Transfer of pollen grain from anther to the stigma of another flower of same plant. That is also a type of pollination. So transfer of pollen grain from the anther to the stigma of another flower of the another plant. That is called what? Also a pollination. So we'll be discussing all these different types. Autogamy, that is called self-pollination. What is autogamy? It is the transfer of pollen grain from the anther to the stigma of same flower. So this is a flower is same, is having anther. From anther, there will be production of pollen grain. Pollen grain will be landing from here to the stigma of same flower. So that this is called what? Self-pollination. In flowers with exposed anther and stigma, complete autogamy is rare. If flowers are opening, there is a chance that pollen grain from some other flower may also land on the stigma. So, in if flower is what exposed, if flower is having exposed anther and stigma, complete autogamy is rare. Sometimes question will be there whether complete autogamy is possible in case flowers are having exposed anther and stigma. The answer will be what? It is what? Complete autogamy is rare. Autogamy in such flower requires synchrony in pollen release and stigma receptivity. Also, anther and stigma should be close to each other. So, for autogamy, what is the condition? There should be a synchrony in pollen release and stigma receptivity. What do you mean by this? So at same time, both pollen grains and this and stigma, they are what? Functional, they are receptive. And anther and stigma should be close to each other. They should be very near to each other. Only in that case, there will be autogamy may be possible. Now, autogamy, like plants like viola, that is called common pansy, or chalice, and camelina produce two types of flowers. This is viola, this is chalice, this is camelina. They bear two types of flower, that is called chasmogamous flower and clistogamous flower. Chasmogamous flower and clistogamous flower. Chasmogamous flower and clistogamous flower. This is what chasmogamous flower. This is what clistogamous flower. These are what clistogamous flower. Now, what do you mean by chasmogamous flower? They are similar to flower of other species with exposed anther and stigma. So, chasmogamous, they are having what? Exposed stigma and anther. Anther is exposed as well as stigma is exposed. That flower is called what? Chasmogamous flower. What's clistogamous flower? They do not open at all. They never open. Anther and stigma lie close to each other. They are autogamous. When anther dehisces in the flower bud, pollen grains come in contact with the stigma for pollination. Clistogamous flower produce assured seed set even in the absence of pollinator. Clistogamy lead to inbreeding depression. What is inbreeding depression? If there is a continuous self-pollination, there will be decrease in quality. That is called what? Inbreeding depression. So clistogamy lead to inbreeding depression. Sometimes question will be there. Clistogamy or chasmogamy lead to inbreeding depression. In clistogamy, there is always Inbreeding. Outbreeding is not possible, so cleistogamy is what lead to what? Inbreeding depression. What is inbreeding depression? Decrease in the quality because of continuous inbreeding, cell pollination, that is called inbreeding depression. Now we'll be discussing on geitonogamy. What do you mean by geitonogamy? It is the transfer of pollen grains from the anther to the stigma of another flower of same plant. This is the plant is same. But here the transfer of pollen grain from the anther to the stigma of another flower of same plant. So that is called geitonogamy. It is functionally cross-pollination involving a pollinating agent. There is an involvement of pollinators. But it is genetically similar to orthogamy since the pollen grains from the same plant. So though it is about functionally cross-pollination, but because here same plant is there, so the genes of both the are same. So they are what? Similar to orthogamy. So geitonogamy is nothing, it is what? Orthogamy. Now third type is gynocim. What happened in this case? It is the transfer of pollen grain from anther to the stigma of a different plant. This is plant A, and this is a plant B. So the transfer of pollen grain from one plant, from anther to the stigma of different plant, that is called what? Gynocim. This is what? Orthogamy. This is what? Geitonogamy. This is called gynocim. It brings genetically different pollen grains to the stigma. So now we'll be discussing about agents of pollination. There are two types of agents, abiotic agents and biotic agents. What is abiotic agent? Wind and water. They are abiotic agents, whereas insects, birds, bats, primates, arboreal rodents, reptiles, and many more. They are what? Biotic agents of pollination. So this is what? Here it is pollinated by wind. Here these plants are pollinated by water. And there are some plants which are their pollination by honeybees, insect, birds, bats, primates, rats, reptiles as well. Now, what is a biotic agent? Like that is called wind. Wind pollinated flower. Pollination by wind is called anemophily. So what is anemophily? And pollination by air, wind, that is called anemophily. Most common abiotic agent, wind pollinated flowers often have a single ovule. They have what? Single ovule in each ovary and numerous flowers packed into an inflorescence. There are numerous flowers. Example, corn cob. This is a corn cob. The tassel are the stigma. These hair-like structure, they are nothing. They are what? Stigma and style, which have the wind to trap pollen grain. Wind pollination is quite common in grasses. Ways for effective pollination. Flowers produce large amount of pollen grains. Here, in the wind pollinated plant, there is a flower. Flowers produce large amount of pollen grains. Pollen grains are light and non-sticky. They are light in weight and they are not sticky. They often possess well-exposed stamen for easy dispersion of the pollen into wind's current. Large feathery stigma to trap airborne pollen grains. Now, secondly, the abiotic agent is water. So pollination by water is called hydrophily. It is quite rare. It is limited to about 30 genera, mostly monocotyledons. Example, Vallisneria. This is Vallisneria. Then Hydrilla, that is present in fresh water, and Zostera. This is Zostera present in what? Marine water, seagrass. This was what hydrophily. But in lower plants, water is a regular mode of lower plants means algae, thallophytes, bryophyte, pteridophyte. These are what lower plant. Water is regular mode of transport for the pollen gametes. Distribution of some bryophytes and pteridophytes is limited because they need water for transport of male gametes and fertilization. So sometimes there is a question is there why biophytes and pteridophytes are present in nearby water bodies because for pollination water is essential. That's why they are restricted to wet area, aquatic area. In Vallisneria, this is Vallisneria. This is Vallisneria. The female flower reaches the surface of water by long stalk. This is what female flower. It is having a very long stalk. And the male flower or pollen grains are released onto the surface of water. They are carried by water current and reach the female flower. The pollen grains. Male flowers are deep inside. They are not at the surface of water. But when they are released, they will be reaching at the surface and then they will be ensuring the pollination. In sea grasses, female flowers remain submerged in water. Pollen grains are long and ribbon-like. They are carried inside the water and reach the stigma. The pollen grains of most of the water pollinated species have mucilaginous covering. So pollen grains are covered with mucilage to protect from wetting. Not all aquatic plants use hydrophily. In most of the aquatic plants like water hyacinth, this is water hyacinth, or water lily, this is water lily. The flower emerges above the level of water for entomophily or anemophily. There is not hydrophily, but there is either entomophily or anemophily. Wind and water pollinated flowers are not very colorful and do not produce nectar. So if you think that if you see that a plant flower is not releasing any nectar, not having smell, so they are not pollinated by what? Insect. They are pollinated by water and wind. Now, biotic agents, animals used by majority of flowering plants, like example bees, butterflies, flies, beetles, wasps, ants, moth, birds, sunbirds, hummingbirds, bat, primates, lemurs, arboreal like tree-dwelling rodents, reptiles, gecko lizard, garden lizard. These are the various animals which facilitate pollination. Pollination by insect is called entomophily. Particularly bees is most common. Often flowers of animal pollinated plants are specifically adapted for a particular species of animal. Features of insect pollinated flowers, large. So the flowers of insect pollinated flowers have some qualities like they are large, colorful, fragrant, and rich in nectar. Nectar and pollen grains are the floral rivers for pollination. Small flowers form inflorescence to make them visible. Flowers pollinated by flies and beetles secrete foul odors to attract these animals. Pollen grains are generally sticky. When the animal comes in contact with the anther and the stigma, its body gets pollen grains. When it comes in contact with the stigma, it results in pollination. Some plants provide safe places as floral rivers to lay eggs. Example, Amorphilus. It has the tallest flower of 6 feet. This is Amorphilus, tallest flower. What is the length? 6 feet. A moth species and the yucca cannot complete their life cycle without each other. This is the yucca plant. This is the moth. They cannot complete their life cycle without each other. The moth deposits egg in the locule of ovary. The flower get pollinated by moth. The larvae come out of the egg as seed start developing. Many insects consume pollen or nectar without bringing about pollination. They are called pollen or nectar robbers. They feed the nectar, but they don't bring pollination. So they are known as what? Pollen or nectar robbers. Now, outbreeding devices. We'll be discussing what is outbreeding. Opposite of self-pollination, that is cross-pollination, that is outbreeding. How plant ensure outbreeding? Because if there is continuous inbreeding, it will result into inbreeding depression. So, how plant ensure outbreeding? What are devices? Hermaphrodite flowers can undergo self-pollination autogamy. Continuous self-pollination result into inbreeding depression. To avoid self-pollination and encourage cross-pollination, there are some devices in plant. This is what self-pollination. This is what cross-pollination. So if there is a self-pollination, it will result into inbreeding depression. Here it will be result into depression. And here there will be, to avoid inbreeding depression, we have to go forward cross-pollination. So there are some techniques. First is what? Avoid synchronization. Here, the pollen is released before, in this method, pollen is released before the stigma become receptive or stigma become receptive before the release of pollen. Second method is arrangement of anther and stigma at different position. Third method is self-incompatibility. Sometimes question is there, what do you mean by self-incompatibility? It is a genetic mechanism to prevent self-pollination from the same flower or other flowers of same plant, from fertilization by inhibiting pollen germination or pollen tube growth. So in case, what is this? In case the pollen grains or stigma or same flowers are coming in contact, the pollen grain will not develop. It will not develop pollen. Yeah, stigma will become what? Non-receptive. This is called what? Self-incompatibility. Next is method for outbreeding is what? Production of unisexual flower. If male and female flowers are present on the same plant, monoecious, like castor and maize, it prevents autogamy but not geitonogamy. Production of unisexual flower if male and female flowers are present on the same plant. It is monoecious, like this castor and maize. It prevents autogamy but not heteronogamy. Dioecious plant like papaya, male and female flowers are present on different plant. This is called dioecious. Sometimes what is the question? What do you mean by dioecious? Term dioecious. You have to remember these important terms. Dioecious means presence and female flowers are present on different plants. This prevents both autogamy as well as geitonogamy. So this is papaya male plant, this is papaya's female plant. Now pollen pistil interaction. What do you mean by pollen pistil interaction? It is the process in which pistil recognizes compatible or incompatible pollen through the chemical components produced by them. If the pollen is compatible, that is right type, the pistil accepts it and promotes post-pollination event. Pollen grain germinate on a stigma to produce a pollen tube through one of the germ pore. The content of pollen grain move into the pollen tube. Pollen tube grows through the tissues of stigma and style and reaches the ovaries. So these are the images shows in pollen grain germinating on the stigma. These pollen tube growing through this style. Then ultimately this they are reaching to what? Fertilized with XII. If the pollen is incompatible, wrong type, the pistil reject pollen by preventing pollen germination or the pollen tube growth. If wrong type of pollen grains has landed, the pistil will prevent the germination of pollen. That is called incompatible. The plant which set pollen grain at two-cell condition, vegetative cell and generative cell. The generative cell divide into two male gametes during pollen tube growth. In plant which set pollen in three-cell condition, pollen tube carry two male gametes from the beginning. Pollen tube reaches the ovary, then enters the ovule through micropyle. And then enter one of the synergate through the filiform apparatus. The filiform apparatus present at the micropylar part of the synergate guide the entry of pollen tube. So this is a pollen grain landing on the stigma. Now it will develop what? Pollen tube. Now pollen tube is having what? Gamete. So this is having one ovule. This is having one ovule. Now here ultimately this uh, pollen tube will discharge its nucleus in once the synergate. Now synergate will guide this nucleus to one nucleus will fuse with XL and that will fuse with a polar nuclei. A plant beetle can manipulate pollen pistil interaction even in incompatible pollination to get desired hybrid. Now, artificial hybridization. Now, what do you mean by artificial hybridization? It is a crop improvement program in which desired pollen grains are used for pollination. This is achieved by following methods: emasculation and bagging. Now, what is emasculation? What is bagging? Emasculation, removal of anther from the bisexual flowers. So this is a bisexual flower. If we are removing the anther, the flower bud of female parent before the anther dices. So after dices, then there is a bagging. Here, emasculated flowers are covered with a suitable bag made up of butcher's paper. This is the bag made up of butcher paper. to prevent contamination of its stigma with unwanted pollen. When the stigma attain receptivity, mature pollen grains collected from anther of male parent are dusted on the stigma. Then the flowers are rebagged and allowed to develop the fruit. This is a simple procedure. This is called artificial hybridization, where there are two steps: emasculation, that is removal of anther, and then there is a bagging. For unisexual flower, there is no need for emasculation. Sometimes question there whether the emasculation is required in unisexual flower. No, answer is no. Female flower bud are packed before the flower opens. When the stigma becomes receptive, pollination is carried out using the desired pollen and flower repacked. Now we'll be discussing about double fertilization. What is double fertilization? After including one of the synergids, here is synergids, the pollen tube uses two male gametes. Here the pollen tube is having two male gametes. These two male gametes will be released here into cytoplasm of synergids. One male gamete moves toward the egg cell and diffuses with the nucleus, so that is called syngamy. One male gamete fuses with the egg cell, that is called what? Syngamy, to form zygote. Zygote is what? Diploid. The other male gamete moves toward the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus pen. Here, so one gamete will fuse with this egg cell, and second will fuse with this what? Polar nuclei. So here already two nucleus are there, and more nucleus, so it will become what? Triploid. So that is called what? Primary endosperm nucleus. As it involves fusion of three dihaploid nucleus, it is called triple fusion. So there is a fusion of three nucleus, one male gamete and two polar nuclei. That's why it is called what? PEN, primary endosperm nucleus. That is what? Triploid, 3N. That word is diploid. So since there is two-time fertilization, that's why that's why it is called double fertilization. One is syngamy and another is what? Triple fusion. Since two type of fusion, syngamy and triple fusion, take place in an embryo site, it is called double fertilization. It is an event unique to flowering plants. So double fertilization is present only in flowering plant, not in gymnosperm, not in pteridophyte, not in rhaophytes and thallophytes. The central cell after triple fusion become primary endosperm cell. So this cell will become a primary endosperm cell, that is PEG, and develops into endosperm. It will develop into endosperm. And zygote develop into an embryo. This zygote will develop into embryo, and this pen will develop into what? Endosperm. So what is post-fertilization event? Endosperm development, then embryo development, ovule into seed, and ovary into fruit. So there is a polar nuclei, then one egg. Here there is step number 2, 3, 4. Then sperm cell, two male gamete, one male gamete sperm cell will produce with egg, and second one with what? Polar nuclei. So egg will develop into zygote, and then this pen with primary endosperm nucleus will develop into endosperm. Then there is a formation of endosperm. Then this zygote will develop into embryo, and this fruit, or it will develop into fruit, and this fruit will seed, there will be seed fruit. So how zygote develop? This is a zygote. Okay. Then it will divide mitotically. It will come to cell stage, and there will be multiple division. And one nucleus will become enlarged. Then there will be sequence of divisions until ultimately there it will be converted, zygote will be converted into globular embryo. Then hard shape embryo, then it will develop into mature embryo. So these are different stages of embryo development. Zygote, then globular embryo, hard shape embryo, then mature embryo. Now, what is endosperm development? Primary endosperm cell, it is primary endosperm cell. This primary endosperm cell divide repeatedly to form a triploid endosperm tissue. Endosperm cells are filled with reserve food material. They are used for nutrition of the developing embryo. In common endosperm development, primary endosperm nucleus undergoes successive nuclear division. There will be only nucleus division to give free nuclei. This stage is called free nuclear endosperm. Number of free nuclei varies greatly. Endosperm becomes cellular due to the cell wall formation. Tender coconut milk, this tender coconut water, is free nucleated endosperm made up of thousands of nuclei and surrounding white canal is the cellular endosperm. So this is what nuclear endosperm, this is the cellular endosperm. Embryo develop a micropylar end. Embryo develop towards what? Micropylar end. The embryo sac where the zygote is situated. Most zygote divide only after the formation of some endosperm. This gives nutrition to developing embryo. Sometimes question is there why there is embryo development is preceded by endosperm development because endosperm is required to provide nutrition to developing embryo. In monocots and dicots, seed differ greatly, but embryogenic, early embryonic development is similar. So the zygote develop into co-embryo develop into globular embryo, then sub-embryo, then mature embryo. Now we'll be discussing about dicotyledonous embryo. It is having an embryonal axis and two cotyledons. So this is one embryonal axis and it is having two cotyledons. These are the cotyledons. So this is a typical dicot embryo. The portion of embryonal axis above the level of cotyledon is called epicotyl. So portion of embryonal axis above the level of cotyledon is called epicotyl. This area is called epicotyl. And which terminate with plumule stem tip, and it will ultimately be converted into plumule, that is called epicotyl. The cylindrical portion below the level of cotyledon is called, this whole area is called hypocotyl. That terminates with the radical, that is, this hypocotyl terminates into radical, and that is radical is supported by root cap. A monocotyledonous embryo, they possesses only one cotyledon. This is for the embryonal axis. They are having only one cotyledon. This cotyledon is called scutellum. It is situated lateral to embryonal axis. At its lower end, the embryonal axis has a radical. Lower end, it is having radical. This is covered by root cap, enclosed in chorioryza. This is enclosed in what? Chorioryza. Portion of embryonal axis above the level of attachment of scutellum is epicotyl. So this area is called epicotyl. It has shoot apex and a few leaf primordia enclosed in coleoptile, a hollow foliar structure. So this is nothing, it is a hollow foliar structure. It protects what? Shoot cap. There is epiblast cell is also there. Now we'll discuss about the seed from ovule. This is the structure of dicot seed. This is the structure of monocot seed. Seed is nothing, it is the fertilized ovule formed inside fruit. It is the final product of sexual reproduction. It consists of seed coat, seed coat, cotyledons, and embryonal axis. This is what embryonal axis. Cotyledons are simple, generally thick and swollen due to storage of food, as in lecture. Cotyledon, they store food material. This is a bean seed dicotyledon, spawn seed monocotyledon. Which have an interest from here. Mature seeds are two types, non-albuminous seed and albuminous seed. So seeds are two types, non-albuminous seed and albuminous seed. Non-albuminous, they have no residual endosperm. Non-albuminous, no albumin, no endosperm. Non-albuminous, no endosperm is completely consumed during embryonic development, like dicots, pea, groundnut, beans. Non-albuminous and albuminous. They contain what some endosperm like wheat, maize, barley, castor, coconut, and sunflower. They are what albuminous. Occasionally, in some seed, black pepper, beet, remnant of nucleus are also persistent. It is called what perisperm. So, what is perisperm? Remnant of nucleus, that is called perisperm. Intrugment of ovule harden a tough protective seed coat. It has a small pore micropyle. So which there is exchange of what gases, oxygen and water enter into the seed during germination. As the seed matures, it becomes dry by reducing water content, 10 to 15% moisture by mass. The metabolic activity of embryo slows down. It may enter a state of inactivity, that is called dormancy. So what is dormancy? The state of metabolic inactivity, that is called dormancy. Under favorable condition, moisture, oxygen, and suitable temperature, they germinate. Now, what is the advantages of seeds? Since pollination and fertilization are independent of water, seed formation is more dependable, but better adaptive strategies for dispersal of new habitat. It helps the species to colonize in other areas. They have food reserve, so young seedlings are nourished until they are capable of photosynthesis. So these are the advantages of seed. The hard seed coat protect the young embryo. Being products of sexual reproduction, they generate new genetic combination and variation. Dehydration and dormancy of the mature seeds are crucial for storage of seed. Can be used as food throughout the year and also to raise crop in the next season. Now, viability of seed after dispersal. In few species, the seed lose viability within a few months. Seeds of many species live for several years. Some seeds can remain alive for hundreds of years. The oldest is that of lupine, Lupinus arcticus, seed, oldest seed. Excavated from Arctic tundra, the seed germinated and flowered after an estimated record of 10,000 years of dormancy. 2,000-year-old viable seed is of the date palm. This is what date palm. Phoenix dactylifera. Discovered during the archaeological excavation at King Herod Palace near the Dead Sea. These are leafless seed, these are phoenix seed. Leafless plants, phoenix plant. Now, fruit from ovary. The ovary develops into a fruit. Transformation of ovule into seed and ovary into fruit proceeds simultaneously. The wall of ovary develops into pericarp. Pericarp is known as wall of fruit. Pericarp is divided into three layers: exocarp, mesocarp, and endocarp. Then inside fruit there is a seed. The fruit may be fleshy like guava, orange, mango, etc., or dry like groundnut and mustard. Fruits are two types: true fruits and false fruit. True fruit, what do you mean by true fruit? In this, fruit develops only from the ovary. Other floral part degenerate and fall off. Example, most plants. False fruit in these, the thalamus also contribute to fruit formation. Example, apple, strawberry, cashew. So it's apple, strawberry, this is strawberry, this is apple. The thalamus, this is nothing, edible part is thalamus. Sometimes question is there. What is the edible part of the apple? Thalamus. Thalamus is part of fruit. It is a apple is a true fruit or false fruit. It is a false fruit. Same way here also. The strawberry, there is a thalamus. There is what? Thalamus. They are called achene. They are called achene. This is cashew nut. This is the fruit. It is nothing. It is the thalamus. In some species, fruit develop without fertilization. Such fruits are called parthenocarpic fruit. Example, banana develop without fertilization. So they are what? Parthenocarpic fruit. Parthenocarpy can be induced through application of growth hormones. Such fruits are seedless. These are the seedless watermelon, seedless pineapple. They are also parthenocarpic fruit. Apomixis. There is now we'll be discussing about apomixis. Apomixis and polyembryo. What is apomixis? Apomixis is the production of seed without fertilization. Some species are asteraceae and grasses. Is that it? Some flower species, they are asteraceae. It is a form of asexual reproduction that mimics sexual reproduction. So, then question is there, name the term which mimic sexual reproduction. So that is what apomixis. Development of epimetric seed. In some species, the plant itself is formed without reduction division and develop into embryo without fertilization. In many species like mini citrus and mango varieties, some nuclear cell, is that what? Nuclear cell surrounding the embryo sac divide, protrude into embryo sac to form embryo. Thus, each ovule contain many embryos. Once occurrence of more than one embryo in a seed is called polyembryo. So there are in one seed there is a single seed, there is many embryo. 1, 2, 3, 4. That is called polyembryo. Polyembryo is reported in citrus and mango. Now, importance of apomixis in hybrid seed industry. If the seed from hybrids are sown, the plant in the progeny will segregate and lose hybrid characters. So production of hybrid seed is costly, so hybrid seeds are also expensive. If the hybrids are made into apomix without sexual reproduction, then there will be no what? There will be no segregation. So there will be no segregation of the character in hybrid progeny. So farmers can keep on using hybrid seed to raise milk crop. So this is all about this chapter. I hope you may have enjoyed this chapter. Your concept may be clear. In case if you are having any doubt, you can share your doubt in the form of comment. Thank you. Thank you very much.